which will be talking about HIV treatment. And so today we have presenting for you Alan McCord, who is the Director of Education at Project Inform in San Francisco. And so, Alan, I'll give you a little bit of time to introduce yourself, and then we will go ahead and get started.
Hello, everyone, and thank you to the AI for inviting me to present this pre-CROI review for you all. I've attended CROI for now, I think, 8 years, and so I'm pretty well knowledgeable about what happens there. And so what I wanted to do with this slide set is to highlight some of the words, phrases, drug names, and that sort of thing to keep your eyes out for during CROI. And those things you can look at in abstract titles, or sometimes you have to dig a little deeper into the text to find these words. Sometimes what may be pre-exposure prophylaxis isn't quite as clear as the name of the abstract presentation. So I'll try to highlight that throughout the slide set, and also give you some background information about CROI and clinical study, and then finish up with some peer-based research. So next slide.
And one thing I forgot to mention, Alan, before you get into your presentation,
Sure.
is if you have questions, please feel free to raise your hand or chat in the question box. And Alan doesn't mind if we interject during his presentation for questions. So please, if you have any questions, just let him know. Okay.
Thank you. Okay, so what is CROI? And you probably already know it's the Conference on Retroviruses and Opportunistic Infections. This has been going on, I believe, for 20 years or more. It is a major global scientific conference, and a lot of the HIV research data that comes out is presented at CROI. Not all of it, but certainly a good deal of it. Over the years, over the last 5 to 10 years, there's less information on opportunistic infections, at least within developed countries, because once you're on HIV treatment, those OIs are less of an issue. So you see much less data on OIs, but you will see a good deal of data on non-HIV related conditions such as cardiovascular disease, liver, kidney disease related with a person taking HIV medications. And then there's more data on implementation and cost effectiveness programs. Increasingly, the last 2 years, as new hepatitis C drugs have come to market, more clinical data around hepatitis C has come out at CROI as well. So those are broadly the kind of data that you will see at CROI. Thousands of researchers, also clinicians and community folks attend. And there are a variety of presentations. There are posters and abstracts. So at the smallest level, there are abstracts that are just a few paragraphs long, which are in the CROI, on the CROI website. There are also posters, which is the next largest amount of information that you can see during poster presentation time on each of those days at CROI. And then there are different kinds of oral presentations, some that last 2 minutes, some that last 5, others that last 15 or 30 minutes. The larger the presentation, the more significant it tends to be the data that is presented in those presentations. So those presentations that are 15 and 30 minutes long, as well as things that are called late breakers, are significant results that you should take note about. And as well, CROI reports on all stages of research. So that includes the earliest animal research and also pre-animal research, just looking at different compounds, then animal study and then human study, which I will also get to in a series of slides here soon. So next slide. So I'm assuming that probably everybody knows about the life cycle of HIV. Sorry, this is the preclinical set. So I wanted to show you the stages of clinical study. So go on to the next. And so it does start with preclinical studies. So let's say that happens in the laboratory before it gets into humans. And this is screening compound laboratory research and animal research in monkeys and rats or humanized mice models. And this could happen over the course of many years, trying to screen compounds and use them in animals to see about their toxicity and ability to control HIV. So that's preclinical studies. Next slide. And then we'll move into human study, and that is phase 1. And this is very few. When it comes to HIV medications, this is really a model that I'm showing you for HIV medication. For phase 1, it'll be a few people, several dozen individuals, and they're usually HIV negative. Only 2 weeks in length, and really looking at safety and a range of doses, and obviously not as expensive as later study happened. Next slide. Then we go on to the second phase of study, and in CROI you'll see preclinical data being presented. You'll also see phase 1 and phase 2, and sometimes you'll see that divided up between phase 2A or phase 2B. So those are some of the words and phrases to look at when you're evaluating the data that's being presented. So in phase 2, we're now looking at many dozens, if not a few, very small 100 number of individuals. HIV positive people then, these are months to perhaps years in length, but maybe 1, maybe 2 years, probably the most that phase 2 will go. There may be a comparison arm continuing to look at safety and dosing, but also effectiveness at this point for phase 2. And 1 out of 3 drugs actually make it through phase 2. So this is a big deal for many of the drugs to get through this phase. Next. So the next phase is phase 3. Many more people. Now you're in the hundreds of individuals when it comes to HIV medications. In other areas of research like vaccines or HPV research, you'll see thousands of individuals in phase 3 study, but with HIV medications you often see hundreds of people, all still HIV positive, usually up to 2 years of data, and you will all often see 24-week, 48-week, 72, and 96-week data come out from phase 3 studies. So again, those are words to look for and evaluate, but most HIV medications final data will come out at 96 weeks. Also, comparison arms here, so the drug, a group of people on the medication that you're looking at and versus a placebo group is what would be typical in a phase 3 clinical study phase. Continuing to look at safety, the dosing has already been taken care of in the earlier phases of study. Effectiveness is really the safety and effectiveness are the two highlighted areas in phase 3, and a great deal more money is spent on this because of the number of people and the length of the phase of study. And next. And after phase 3, then, if it's a promising result for this clinical study, then the company that has been developing that drug can present it to the FDA for either approval or denial. So that usually takes up to 6 months and sometimes 12. If it's a game changer as far as medication is concerned, the approval or decision may happen within 3 months. Then there is a fourth phase of study that we often don't hear about, and at CROI you're never going to see someone saying this is phase 4 study. But this is post-FDA approval, continuation of following those individuals on this particular compound. And this is thousands of individuals, a great deal more money that's being spent, but it's collecting information on real-world experiences versus the clinical trial setting. So it's really collecting long-term data on the effectiveness of this drug. So within the 2 years that you see in phase 3, sometimes those long-term side effects may not appear until year 4 and 5 and 6. So it's good that some phase 4 study does happen, but it doesn't happen that often, and the FDA does not enforce this as much as what I think it should. So that is the basic, 4 phases of clinical study in humans. And so just beware as you're looking through CROI data. They will often say it's preclinical, phase 1, phase 2, and phase 3, and so you'll have a sense of where it gets placed in the long run. So next. So I'm not going to go over the life cycle, but this is the life cycle of HIV. I'm assuming that most people probably already have seen this, but I did want to use this as a basis to point out the various targets for HIV medication. So next. And the first target is going to be on GP120 on HIV. This is the latest class of medications that are being looked at over the course of HIV treatment investigation. So this is one area to look at, GP120 inhibitor. So next. The next step in the life cycle is attachment to the CD4 receptor. So look for keyword CD4 inhibitor or CD4 receptor or attachment inhibitor if you're trying to follow this class of medication. And there is one that's being investigated. Actually, there's one drug in both of these classes that there should be data at CROI on. Next. And the third area is the R5 co-receptor. So we look for R5 inhibitors or CCR5 inhibitors. And again, there is a study on R5, new drugs in the R5 inhibitor class. I will go over this in a slide later to explain what those drugs are. So next slide. And this is the fusion step. And so I don't recall right now if there is a fusion inhibitor that's being studied. But if it is, then you can either look at fusion inhibitor as the keywords for that class of drugs. Next. This is probably what a lot of people already know about is NRTIs or NNRTIs, nukes and non-nukes. This is the reverse transcription step in the life cycle. And obviously we have 2 large classes, nukes and non-nukes already, and there is a new NRTI as well as a new NNRTI that you will see data on at CROI. Next step, or next slide. This is integration, and there is a new integration inhibitor. And so look for those words, integrase inhibitor. Next. You see this is the protease, the transcription where the protease enzyme is part of that process, and so protease inhibitors. I doubt very much that there's any data on any new protease inhibitors. I don't remember anything. If there is, it's very, very early study for new protease inhibitors. And then the next 2 slides, you can move on to those 2. And these are maturation inhibitors. Maturation inhibitors have been studied for a while. They have come short on being able to be approved by the FDA because they just don't come up with the same level of viral suppression as other classes of existing drugs. But there may be 1 or 2 compounds that are talked about around maturation inhibitors at CROI this year. So next slide.
Alan, we did get a question for you, and when you were talking about the the research cycle, can you explain what you mean by a blinded and double-blinded study?
Yes, blinded and double-blinded. And there's also unblinded. So there's many different aspects of clinical study, and the gold standard would be a double-blinded study. But let's go back to unblinded. Unblinded would mean that everybody knows who's taking what. So that includes the researchers, knows who that drug is given to, and then all the participants know what they're taking. So they know that they're getting a placebo or they know they're getting the drug. So blinded would be when the participants don't know, but the researchers do know who's getting what. And then double-blinded is nobody knows who's getting what. That actually excludes a lot of bias in the clinical process, clinical study process. So that's really the gold standard, and you will almost always see, at least in phase 3 studies, double-blinded, placebo-driven studies in order to remove as much bias as possible.
Thank you.
I hope that answered that question. If not, let me know. So just a wrap-up from that life cycle. So you have these 5 current classes of medications, entry inhibitors. You will also look, again, as I said, look for attachment inhibitors, R5, CCR5, or CD4 inhibitors as words to look for. You also look at the second class, which is nukes and non-nukes, NRTIs and NMRTIs. Third group are integrase inhibitors, and you will see this written in several different ways, either integrase inhibitors, INSTIs, or INIs, I-N-I-S. So any of those will apply to integrase inhibitors. The fourth class, protease inhibitors, and then the fifth class, maturation inhibitors. So next. So wanted to go over the medicine cabinet, as you will, what's currently approved by the FDA and on the market. And as you can see, those 5 classes, entry inhibitors, NMRTIs, INSTIs, protease inhibitors. Then you also have these other 3 groups, fixed dose combos, which is 2 or more medications together in one pill that are not full regimens. But then when you have 3 or more medications in a single pill that is a full regimen, then those are called STRs, or single tablet regimens. So that's another acronym to look for at CROI is STRs, and there currently are 4 on the market. And then there are boosters, those drugs that are used in order to boost a few of the main medications in the regimen. So next slide. So this is the full medicine cabinet, although there's 2 other fixed dose combos that I haven't added there. And so in total there's 37 medications. Now, not everyone is going to go through those medications. And you may see data on some of these as far as that phase 4 sort of follow-up study. But by and large, these are the things that was presented at CROI over the years with a great deal of research data. So, and so this is the sort of information that you'll see on the new drugs that are coming out. If you go to the next slide. This next slide grades out some of those drugs that people new to HIV medication who are going on it for the first time will likely not take. Someone's not going to take Fusion because that's an injected drug twice a day, so that's not something that somebody would want to go on for the first time. There are other drugs that are very old and problematic as far as either viral suppression, adequate viral suppression, or the number of side effects and the kind of side effects that they have. And that would be the Xerit, Restrictor, Viramune, Crixivan. Those are older drugs, and since we have newer drugs that are a lot more tolerable and a lot better suppression of HIV, then those are the ones that we'll see being used a lot more often. So next one.
And we have another question.
Yeah, sure.
So can you explain the role of, I don't know how to say it, combisistat?
Combisistat?
Yes.
Yes. So as you see the 2 boosters down there, and I have ritonavir, it's also called Norvir. That's an anti-HIV medication. Combisistat is the first non-HIV medication that's being used in certain combinations. So ritonavir over time has been fairly problematic for a lot of people. It's certainly given a full dose. Ritonavir has a very long list of side effects. When it is used as a booster, it's either 1 or 2 pills every day. Far fewer side effects, but it's still somewhat problematic for a good number of people. So ritonavir is used with protease inhibitors. So you'll see that boosting Persista, Reataz, Kaletra, and Lexiva. So there's been desire over the years to have a non-HIV or a replacement for ritonavir, and that's where Covisistat has come in. Covisistat is also called Tybost now on the market. So Covi is used in one STR called Stribild. If you look up there, it says Elbitagravir, Covi plus tenofovir and FTC. So Covi is a non-HIV active boosting drug, and it does boost certain protease inhibitors as well as a couple of the integrase inhibitors. And so it's important that there's now an option to not using ritonavir and trying to reduce the number of side effects over time for some individuals. So now we have two choices rather than one. And if I didn't answer that well enough, then please let me know.
I believe it did. So I'm going to go ahead and move on to the next slide.
Yes, please.
So basically, it's a replacement for ritonavir with fewer side effects, essentially.
Fewer physical side effects. It can, Covisistat can raise a kidney function estimated, it's called eGFR, estimated GFR, glomerular filtration rate. Although that may indicate some decline in kidney function, it appears that it's either a temporary thing or it really doesn't cause ongoing kidney dysfunction. So there is a concern, a slight concern around Covisistat, and I wouldn't quite characterize that Covisistat is a better drug over ritonavir or necessarily a replacement for ritonavir, but I think in practice it may eventually become that because now Covisistat has been combined with two protease inhibitors and it's on combo pill, which we'll get to in another slide later.
Okay, awesome. Thank you for that clarification.
Sure. So this is the timeline. I just wanted to show you, how HIV medications have been developed over the years. And back in 1987, AZT was the first drug, and it took another 4 years until the next drug came on the market. And you see an awful lot of the NRTIs over time there. And then protease inhibitors had its golden era from 95 through up to 2006. And then you see the development of integrase inhibitors, entry inhibitors, and then the various combo pills over time. And so I just wanted to give you context of how these medications have come to FDA approval. Some of those drugs have a strike-through in it, and that means that those are no longer used for HIV either because they're too toxic or they've been replaced by new versions of the same drug. So next slide. So I wanted to give you an overview of what kind of combinations that you'll see at CROI. And basically, when people go on treatment for the first time, it really is Nardi backbone plus one other drug. So minimum HIV treatment, and you probably already know this, is 3 drugs from 2 different classes. And the great majority of those people going on treatment for the first time, you're going to have that Nardi backbone or 2 NRTIs, which would be Epsicom or Truvada, plus 1 drug from another class of drugs. So either protease inhibitor, integrase inhibitor, entry inhibitor, or an NNRTI. So this is just the basic HIV regimen for most people. Next slide. But you can also have various other treatment regimens as people are switching off from the first and second or third regimen that they're using. Additional medications may be needed. So as in that first example, you'll add an integrase inhibitor to the protease inhibitor. You may continue to have that Nardi background, but now you'll have 3 other drugs from 3 other classes, so that's 5 drugs total. And sometimes there is what's called Nardi sparing regimens, like that last example that you see, which could be 3 or 4 drugs from 3 or 4 classes. without NRTIs being used. Sometimes this happens because people become resistant to certain classes of medications. NRTIs is no different, and so sometimes people can't use NRTIs anymore. And so you'll end up seeing a situation like the last example on this slide. And next slide. And then combinations that won't be used. And 3 NRTIs we don't see all too often. There is a drug back on that one page. It's called trizavir. It's 3 nukes in 1 pill. That is an older combination pill, and we just don't see that being used, certainly not in first-line regimens. So occasionally you will see the 3 NARDIs together, but by and large, at least at CROI, you're not going to see somebody trying to, some researchers using 3 NARDIs in their regimens. You also not see 2 drugs from the other classes, like protease inhibitors, integrase inhibitors, and NRTIs. This greatly increases the risk of severe side effects when you have two medications from these other classes of HIV drugs, which is very different than using 2 NARDIs, which seem to go hand in hand pretty well. So next slide. So, why the need for more medication? So we're going to CROI, why do we need more data on new compounds that are coming out? So even though there's 37 medications, people do not have, unending opportunity to switch to regimen after regimen after regimen. At some point, they're going to stop. You're going to have a limitation on how many medications a person can use over time. Also, if there's more interruptions within the life cycle, then there's better control of HIV. So the new CD4 inhibitor or the GP120 inhibitor are new classes of medication that we haven't seen before. So if somebody, especially in a salvage situation, who has few opportunities for new medications, going to these new classes will help stop HIV from its life cycle in different places than the core classes of medications that we have on the market today. There are also improvements in current medications, and so let's reduce the short and long-term side effects of certain medications. And so there is tenofovir disoproxil fumarate, TDF, which is probably going to switch over to what's called TAF, or tenofovir alafenamide. And there's more information I have on that in a second. Also, improved dosing schedules, monthly injectables or quarterly injectables, improved pill count. If you can cut down a pill once a day, one more pill once a day, that helps with adherence. Also, fewer drugs, fewer side effects, especially in aging. When we have polypharmacy issues, then we want fewer toxicities with these HIV medications. Also, new medications may be developed to overcome class and multi-class drug resistance. The resistance is still an issue. About 14% of Americans living with HIV have some level of resistance to HIV medications. So this is important to develop new drugs around that. And then, obviously, trying to find a functional cure. So those are many of the many, but not all of the reasons for needing more HIV meds. Next. So these are the most recently approved HIV medications, and that medicine cabinet slide did not include the top 2 here. So this would be 36 and 37 medications that have been approved. So Pezcovix and Evotaz were approved last month. These are both combo pills. And to follow up on the cobicistat question, both of these protease inhibitors, the Resistin and Reataz, are combined with Tybost or cobicistat as a combo pill. So instead of taking, I believe, 2 pills, the one Resistin, one that's cobicistat or ritonavir, as it used to be, now it's just one combo pill. for these two drugs. And then you see the rest of the medications over since 2008, the different ones that have come to market. 1, 2, 3 of them have been STRs, Complera, Stribild, and the latest is Trimet from last year. So next one. So in the pipeline, and these are things to look at, when you're reviewing CROI information. So I just crossed Prezista and the Tybos combo pills off because they were approved last month. Again, as I said, tenofovir alafenamide TAF to replace the TDF. So this is a small molecule of tenofovir. The hope is that it has less effect on kidney function as well as bone density. So there is plans, as far as what I know, to have a Stribild version with TAF. It seems like there may be two versions, one with TAF and one with TDF. I'm not sure how that's going to play out as far as one replacing the other over time. But look at TAF as an updated to be included in new regimens as we move forward over the next few years. Doravirine is a very promising new NNRTI. You'll see it as phase 3. I think maybe it might be 96-week data in treatment-naive individuals. So this, I think, has some overcome some mutations, resistance that have been developed of other NNRTIs. So look for doravirine. That is the generic name, phase 3 study. The R5 or CCR5 inhibitor, cenipriviroc, is also in phase 3. I think it might be either 48 or 72-week data that is being presented at CROI. It also is a promising new R5 inhibitor, not only R5 inhibiting R5, but also the other co-receptor R2, which helps reduce inflammation within the immune system. We don't know the effects of what that means, but at least as far as preventing HIV entry through R5, it seems to be a very promising drug. An integrase inhibitor called cabotegravir is also being, you'll see more information on this. This is very early study, maybe for phase 1 data that could be phase 2. I think it might be phase 1. Sorry, I think that might be a mistake on my part. But this is a long-acting injectable, and not only for HIV treatment, but also HIV prevention. So look for cabotegravir for both of those applications. There is a new area of interruption of the HIV life cycle with the GP120 inhibitor called fostemsavir. So this is the first of the kind. It is exciting to see this going through clinical study, and you should see phase 2 results of some kind on this compound. The last two, fastinavir and ibalizumab, are both have been studied. I have not heard much as far as good data regarding both of these compounds, so there may not be any data on these at CROI this year. But if you do see it, then at least you have those listed here on this slide. Next. So, just to give you, one single slide of what to look out for at CROI. So, very exciting news, I think, will be coming out of two PrEP studies, one called PROUD and one called IPRGay. PROUD they're both European studies. PROUD was done in London. IPRGay is done in Montreal and some sites around France. Both of these are looking at effectiveness of PrEP in gay men. PROUD is within the setting of a clinical or sexual health clinic, so individuals asking for PrEP in that context. And then IPRGay is a before and after pill. So the process there would be taking 2 doses of PrEP or Truvada before an expected sexual activity, and then taking 2 doses afterwards. So both of these studies were stopped early last year, I think either August or September, because interim results showed a very high level of efficacy for using PrEP, and I think the leaked out data was more than 80%. So if it's that high, it's really positive results around pre-exposure prophylaxis. So definitely I would think that these would be in late breaker presentations at CROI. So proud and IPRGay, PrEP, Europe, France, London, those are the things to look for there. Again, I said Tenofovir alafenamide, TAF, so look for data around that. Doravirine, again, I said that. Pre-CROI, cabotegravir. So as I said, cabotegravir is not only for HIV treatment, but also HIV prevention. If it's not listed as cabotegravir, the other thing to look for is GSK744. So that's the actual compound for use for PrEP. So for cabotegravir, for treatment, it may be once a month injections. For PrEP, I think they're looking for quarterly injections at that. Both of those would be very early phase 1 or phase 2 study. Fostemsavir, again, mentioned that before. Maraviroc also is being studied for PrEP, continuing to be used as a pill, so look for information around that. Selzentry is the brand name, although brand names are not routinely used at CROI. We'll also look at maintenance therapy, so switching people off an established 3-drug regimen after stable virus suppression for a certain period of time. I'm not, that could vary. It could be 6 months, a year, 2 years of virus suppression, and switching them to a 2-drug regimen. So that may what would help. That would help with one less compound in the body, but also help reduce costs over time. So look for maintenance therapy or switch studies at CROI. Monotherapy, there have been earlier studies that show that monotherapy really isn't a good concept. It could be that we just don't have the right drugs for someone to be on 1-drug regimen, but there may be data on monotherapy at CROI, and then some cure-based research as well. So next. And then what to look out for CROI related to PrEP. Again, PrEP and IPRGay, cabotegravir, you see all those listed there. It didn't also raltegravir, the integrase inhibitor, rilpivirine, the NNRTI as an injected compound. Look for PrEP related to using it as pills, as injectables, as vaginal rings, films, and tablets, and also as vaginal and rectal gels. So that's a very large number of compounds and methods to get it into the body. So keep an eye out for those words and those types of studies, not only in monkeys, not only in humans, but also in early monkey studies. So next. So this is a great graphic by ABAC, and it shows you the very, very deep pipeline for PrEP. Only 1 in phase 4, the Gilead Truvada for PrEP, but 2 now in phase 3. So you may see some information around PrEP in these compounds and methods at CROI. So it's a great graphic. There's a lot of data here. So study this as much as you can and look for those compounds at CROI. So next. So I wanted to talk lastly about cure research that will more than likely be presented at CROI. So just wanted to go through this process of, an HIV negative person getting HIV and then becoming cured, and what that process entails. So here's an HIV negative person, and so next slide. And then within the first 3 months, you have this acute infection with high levels of viral activity, reproduction, and at this point, obviously, people are highly infectious. Next slide. And then after a certain amount of time, the immune system corrects that, gets HIV under control, and becomes to a level of what's called a viral set point. This happens over the subsequent next year. People are still somewhat infectious at this point. And then next. And then this next slide is when somebody goes on treatment and gets their viral load down below undetectable levels with below 50 copies. And as you probably know with HPTN of like 2 study that an individual who's taking medication is 96% less likely to be infectious. So this information you probably all know, but let's get to the functional part in the next slide. So at this point, we can add new compounds to maybe the existing HIV regimen or in place of the regimen. And what's been studied, and these things that these words that you want to keep an eye out for, ZFM or zinc finger nuclease, and HDA-CIS or HDAC inhibitors. Both of those more than likely will have data on different clinical studies that have been ongoing in those areas. Also, valproic acid, therapeutic vaccines, and toll-like receptor 7. There may be other things beyond these 5 things that I've listed here, but these are the things to keep an eye on. HDAC inhibitors, just to let you know briefly, so the viral reservoir are those HIV-infected cells that are resting cells within the body. That's one of the reasons why HIV infection is a chronic condition. As those resting cells reawaken, then new virus is being produced. So HDAC inhibitors actually target those resting HIV-infected cells and perturb them in order to wake them up, and hopefully other compounds can be used to then maybe intensifying HIV treatment while HDAC inhibitors are being used, so that that extra drug that extra HIV medication can suppress the new HIV that's being produced from those perturbed cells. So it's a very interesting area of research. It's still fairly early within that time, but it's very promising research. I really do like following it. So the next slide. So that's the functional cure. This is the sterilizing cure, and you probably know of the Berlin patient who has been cured, and that's a sterilizing cure. He had gotten a stem cell transplant. So I don't know if there's going to be data around stem cell transplants, maybe, but also zinc finger nuclease is a possibility to see data around that as well as far as the sterilizing cure. Then the other last way that we may see more data on, so next slide, is the application of HIV medications during acute infection. So things to look out for here is Visconti cohort from France. And this is a group of individuals, I think it's 65 or 70, that had gone on treatment very early in their infection, within 3 months or 1 month of getting HIV. And over time, for some reason, for various reasons, those individuals have stopped taking HIV medications, and there has been data already produced that shows, I think it was 17 or maybe 20 of those individuals have not had a viral rebound even though they've been off their HIV medications for more than a year. So this is very interesting research. It's a functional cure because it does this process didn't eradicate HIV, but it's similar to a long-term non-progressor or an elite controller where the immune system is controlling HIV on its own after starting HIV medication very early in acute infection. So again, look for Visconti cohort, early treatment, acute infection. Those are some of the cohorts to look for regarding this possible functional cure strategy. And I think that's my last slide. So if you want to go to the last, I think that's my final.
It sure is. And so
yes.
I just wanted to, before we get into questions, I wanted to thank you, Alan, for your presentation. It was very clear and very helpful, even for me. And so I'm assuming that it was helpful for everybody else that was on. And so now everyone will be the time to, if you have a question, to type it either in the question box or to raise your hand, and I will unmute you, and then you'll be able to ask your question to Alan. So while those are coming in, first I had a question about what you thought that the delegates should be on the lookout for CROI, but you already answered that, so you're a couple steps ahead of me. But then I also was thinking along those same lines, since the point of this program is to bring this information back to the community, what do you think some of the most important things that the delegates should bring back to their communities regarding the information that they learn at CROI? So which new drugs, which new prevention technologies should we be thinking about bringing back?
I think on the one hand, PrEP, the vast amount of investigation into pre-exposure prophylaxis, I think there's still not enough education around PrEP. We only have TRUI currently, and so if we diversify that with different strategies, that some individuals may prefer one regimen over another. So to not just talk about the excitement that's around the new research, but also to educate communities that there is this HIV prevention method, and it's very possible within our lifetime that we can stop, pretty much every infection that happens if we reach those individuals. Another area, just generally, like the new medications, maybe not every of the new medications, but that there's these 3 or 4 new meds that are being added to the toolbox, and especially for people in late stage in disease with maybe salvage situations, that the GP120 and the CD4 inhibitor classes are additional interruptions to the step of the life cycle, and so that hopefully offers some hope for those people in salvage situations looking for new regimens. What else? I think maybe some information around cure-based research, that it's happening. There is a significant investment in cure-based research in the United States. It is still in its fairly infancy, but there's promise there, especially around if we can get people on treatment as soon as possible, that maybe we can produce more individuals that can go off treatment at some point, much like some of those individuals in the Visconti cohort. All that information helps with, get tested, know your status, make decisions around treatment, and hopefully that you can access that treatment.
Awesome. Thank you. And we did have one question that came in from Danielle, and she was asked, we have another one that just came in, but the one from Danielle said, can we really equate a sterilizing cure to a complete eradication of the virus? So do we know that, for example, in the Berlin patient, is there no more HIV in his body at all? Do we know that, and can we really say that that's completely a sterilizing cure?
As far as what I've read, the last data that's come out, it is believed that he is free of HIV. There has been, I believe there were 2 labs that reviewed blood samples. One of those labs, I think, found fragments of HIV, and the other lab didn't, but it didn't appear to be replicating HIV, and it could be an issue of contamination from that process from that lab. I haven't seen data on the Berlin patient maybe within the last 1.5 to 2 years. So if I haven't seen it, I apologize for not being able to tell you more about it. But the last data is around trying to confirm with outside sources whether or not Timothy Brown is indeed cured. And it does appear now, I think 6 years later, that he is cured. So it's something to, when we say the word, it's a pretty powerful word, but we, from a scientific point of view, I think we really want to make sure that that's the case. But as far as what the data is so far, it does appear that he is cured. And then the other question you had?
There was one that was actually a question for me. But so as we were talking before the webinar started, Alan, some of the delegates are going to be hosting post-CROI updates in their communities, and so the question was for me about whether I will be compiling a slide set. And I'll give you all a template, but you'll be responsible for filling in that information. And we'll talk a little bit about that some more when we're actually in Seattle, and kind of talk about what those slide sets will look like for your community updates.
And let me piggyback on that. If you'd like to run that by Project Inform, we'd be happy to add in anything. David Evans, who is our Director of Treatment Advocacy here at Project Inform, he is attending as well as I'll be there, so we can help with that if
awesome.
you want that second pair of eyes.
Yes, absolutely, and I'll send you both actually an invitation to our breakfast updates, so that you all, if you have the time, can come and talk to the delegates as well. That'd be great. So it doesn't look like we have any more questions, so I'm going to go ahead and end the webinar. I just wanted to remind everyone who's on the call to make sure that you watch the recording from last week's webinar if you were unable to attend, because it talks about a lot of important logistical and programmatic details for next week. And I'm really excited to see you all again and meet you all, the ones who I haven't met already, and get this pilot of this community education program on the roll. So thank you all for attending, and again, thank you, Alan, and hopefully we'll see you in Seattle.
Yeah, definitely. Yeah, good luck, everyone, and if you do want to get in touch with me, just email me. But it'll be an exciting week for all of you, and hopefully you'll feel the same once you get there.
Awesome. Thanks, everyone.